Hello, thank you for joining us for this webinar. My name is RJessup. I am the perennial grass breeder in the Department of Soil and Crop Sciences at Texas A&M University, and I'm very thankful to have been asked to participate and present some of our work, some of our research in our tropical grasses, and more specifically related to their performance in seed production metrics. And I'd like to start today and really focus on just a part of our research and some of our crops that are more relevant, hopefully, to this audience. And with that, I'll start. And again, I'm going to try to focus on our work in both sorghum and pinniseedum. And in both of those cases, we're working with wide hybrids or crosses between two different species within those taxa. Okay. And our program again is a bit broader. We focus on perennial grasses within the context of biofuels, forage, turf grass, ornamentals, and all manner of renewable bioproducts. And the products that we develop are the feedstocks are really adapted throughout subtropical regions. You can see the southern one-third of the United States, uh, throughout the equatorial regions, and well into portions of Argentina. Okay. And again, I'm going to focus on sorghum and pinniseedum. Within sorghum, I'll visit about two different types of crosses, one with sorghum halopense or Johnson grass, and one with another wild perennial species, sorghum propinquum. And we'll talk about them at different ploidy levels and different end uses. Our work within pinniseedum is really focused on one hybrid crop, and that is between pearl millet, a cultivated crane crop, and Napier grass, to make a sterile triploid crop that is very unique in its production potential. Okay. And in both cases, I'll try to come back to issues that are always occurring in our efforts to make these viable seeded products, and there really are reduced fertility or the potential for reduced fertility in the seed, uh, as well as the very small seed size compared to corn or sorghum or typical grain crops. Okay, so starting with sorghum, again, we cross these, your typical grain sorghum or sorghum bicolor, with two different species, halopense and propinquum, and we look at them at different ploidy levels in the case of propinquum within those. And if I start with the crosses between bicolor and halopense, we've tried these for the longest period of time. We end up having hybrids with acceptable forage quality, yields, and high below ground yields, which are important for carbon sequestration or soil health. Uh, we've released one of these lines that has very high biomass production above and below ground. And we've confirmed the below ground biomass potential of this specific hybrid with ground penetrating radar. And this is a depiction of the above ground trough that was full of sand that we washed away, and you can see all of the exposed roots and rhizomes that are remaining that account for that very large below ground biomass potential that can permeate over time. And we've confirmed that this does contribute to soil organic carbon or long term soil carbon sinks, reaching 1% in less than 5 years compared to 15 or 20 years for conservation tillage systems. Okay. Uh, the limitation of this hybrid, especially in the context of seed, is that it is actually 100% seed sterile. It is an aneuploid between bicolor and halopense, and it must be propagated from rhizomes, and that has significantly limited its commercial appeal and its commercialization to date. So in that system, we've tried to work with seeded types as well. Uh, our efforts have been very similar to previous work, and that crossing sorghum bicolor or sorghum halopense to develop seed is very difficult. They are rare hybrids, get a low percentage recovered, and those hybrids tend to have low quality seed that is not viable. And those that are viable, again, are a very low percentage, 1% or less. So, very low odds of that ever being viable commercially as well between those two. And part of that reason is that the meiotic events result in varied ploidy level in the hybrids and the seed you do recover. Some can be tetraploid, some triploid, and many are aneuploid. So as a result of those issues, we have really transitioned away from working with sorghum bicolor crossed with sorghum halipens. We are now focusing more on sorghum bicolor crossed with a perennial sorghum propinquum. Both of these crops are diploid, 20 sets of 20 chromosomes, so there is no real issue with developing triploids or aneuploids. And we have looked at these both crossing the propinquum with diploid sorghum bicolor and tetraploid sorghum bicolor. We've done these uh, in Texas, and we've developed populations that have very large amounts of genetic variation for us to select upon. You can see variation here in height, flowering time. Uh, we also have variance in rhizomes and all sorts of performance traits. Our breeding efforts have advanced to the F3 generations, and again, we're focusing on rhizome production, perenniality, biomass and forage yield, seed size, and carbon sequestration. And to date, the diploid crosses with bicolor and propinquum have made progress with rhizomes and perenniality, but they have not yet produced a very large seeded hybrid. Okay. Uh, and as a result, we've tried to increase the ploidy level of those hybrids, induce doubling to get to a tetraploid, uh, and we have achieved increased seed size going from an average of 1 milligram from the hybrids at the diploid level to anywhere from 10 to 15 in our new hybrids, and comparing that to sorghum bicolor, which is typically anywhere from 15 to 30 milligrams. Uh, the tetraploid, induced tetraploid hybrids we have from these are much larger in seed size and in reality a bit stronger in perenniality as well. But looking at these in more detail, we have looked at pollen stainability and seed set. Okay. And fertility in seed set is somewhat reduced, not significantly, uh, or not to an extent where it can't be a commercial hybrid. But if you look here, our tetraploid hybrids in general have about a 30% reduction in pollen stainability and seed set. We can still achieve greater than 1,000 pounds per acre. We've reached almost 2,000 pounds per acre seed with this equivalent. So we think it is a viable commercial hybrid, and we think we can improve it with more selection and advancing this through recurrent selection or population breeding methods. Okay. We've also looked at the meiotic segregation in these hybrids, and we have confirmed that, as we expected, both parents, the bicolor and propinquum, have normal meiotic segregation, 10 pairs. The diploid hybrid, full fertility, 10 pairs. The tetraploid hybrids do have one or two occasional quadrivalents or associations of 4 chromosomes, and we believe that has something to do with the reduced fertility in the hybrids. But again, not a barrier that we cannot overcome or work with commercially. Okay. In addition to sorghum, we've worked with pinacetum, and with this I'll focus on one crop, and that involves pearl millet or Pinacetum glaucum, drought tolerant, tropical grain crop that we can hybridize with Napier grass, a related species that actually has 2 genomes in it. One of those genomes is identical to the pearl millet genome, so you end up with a hybrid that has 3 sets of chromosomes, 1 for millet and 2 for Napier, bearing in mind that one of the ones for Napier came from a progenitor similar to pearl millet. And it lets you have hybrids uh, that can be produced fairly frequently, very easily. You have a higher seed set or fertility production in the pearl millet parent when it is crossed with pearl millet Napier grass or with Napier grass. Okay. And in a forage context, this puts us in a nice position where if you look at the red box, At the triploid level, where you have the 3X, you have very high biomass potential for forage, and you have almost no fertility in that triploid seed that's planted. So within the United States, uh, this reduces or removes completely the risk of the plant being invasiveness or spreading by seed. You can produce this hybrid crop from two seeded parents that are fertile, produce a triploid seeded hybrid, sell that seed to producers, and they produce that F1 triploid hybrid in their field, and it's sterile. So one, it's not invasive, and two, they return to you for more of that seed in successive years. And it's very synonymous in a system to what's been done for triploid sterile crops in grapes, watermelon, banana, and other crops. Okay. And for our work in bioenergy, we've really focused on this as, again, being a sterile F1 hybrid system that has high yield potential and can be harvested in year one, which is critical for the commercial viability of forage and biofuels crops compared to some others that have been proposed, switchgrass and miscanthus in particular. Okay. Within our efforts in developing the PMN system, we have developed pearl millet that does not possess one or a few basal tillers. We have some that possess over 100 basal tillers per plant, so profuse tillering. That accounts or that attributes biomass density in the hybrids. And we've also put this idiotype or this type of plant into a male sterile cytoplasm. So we can now use that as an AB line, just as they do in sorghum and other crops, as the maternal or female parent. Okay. We've also worked to improve Napier grass by developing inbreds, and it's been a slow process, but we've gotten to the point of having fourth generation or S4 inbred Napier grass that still has acceptable yield, flowering, and pollen shed potential to be used as the paternal or male parent in crosses. And by doing this, we now have uniformity or inbreds in both parents. The pearl millet is inbred, and the Napier grass is now inbred, which is novel to our knowledge for the species. So we now have PMN seeded triploid hybrids that are uniform in the field. Okay. We've also worked with seeded experiments of PMN identifying chemistries and herbicides that are safe to be used within the crop, and we've identified methods for eradication of the crop, and we've looked at some seed safeners, concept or fluoxethium in particular, as far as rates and effectiveness that are safe in the crop in seed coatings. Okay. Seed fertility in PMN is not an issue. You can develop 100% seed set on the pearl millet parent with a Napier grass pollen. However, the seed you generate is typically small. We've made some progress, but it's always between 1 and 2 milligrams per seed at best. And this translates to being a small seed. And in situations where you have seed that are planted too deeply, or you have heavy soils, high clay content soils, or salt crusting on the surface of the soils, you have significant reductions in germination and emergence on these crops. So seed size has been a large issue with PMN moving towards commercialization and acceptance by producers. Our approach, since we have not been able to make very much progress genetically, has been to pursue hill seeding or seeding 3 or 4 seed at every location in the field where you put a seed as opposed to putting a single seed. And we've expanded that in the, not just seeding hills, but seeding hills that are coated. So at that point, you can add your fungicides, your fertilizers, your other protectants as well. Uh, so we have succeeded making simple clay coated incorporated pellets that have 3 to 4 PMN seeds in them, and when they germinate as a cohort or as a group, they have much better emergence in the field. Seed depth is still an issue, but crusting and clay content have been largely overcome with this approach. Okay. We've also worked to try to improve performance of the PMN seed and seed pellets by priming the seed first. And seed priming is simply a method of partially rehydrating the seed and allowing it to become metabolically active. And then before it actually germinates and the radicle emerges, you dehydrate it back down, and you end up with a seed that is primed and ready to germinate more quickly, and it tends to have genetic responses to whatever stresses you might have placed upon it. We have used this using solid matrix priming or calcine clay, and this allows us to get the moisture content to about 30%, 25 or 30% moisture. It initiates early metabolic activity, but prevents any germination of the seed or the radicle. Okay. We have shown that by inducing priming in combination with different stresses, heat, drought, and salt, you can induce responses in those seedlings that are maintained when it is planted in the field and germinated under those conditions. And we've seen higher yields in the seedlings using solid matrix priming, shown here as the S&P in green, as well as other chemical stressors we've applied, chitosan, azacitidine. Those significant increases in yield, and the most simply using solid matrix priming. Okay, and those seedling yields for establishment have been raised roughly 40%, which in a perennial crop is critical. Right? Okay, this is the sum of what we've achieved in seed quality and seed production in both our perennial sorghums and our perennial millets. And I welcome any and all questions or feedback or comments. Thank you.